Okay, I think we can start. So welcome everyone to today's session of the Plant Cell Atlas Cell Identity webinar series. During this series, we are discussing how we can identify and define cell types and states in the era of single cell approaches. So far, we heard a lot of great talks about cell types and states. And today we will talk about cell types and states during development and also get a glimpse into proteomic approaches to define cell types. This is just in the middle of the seminar series. There were 3 already, and there will be 3 more. And in the very end, we will have an open panel discussion also. But before we start the presentations, I would like to highlight 2 upcoming meetings. The first is the AG2PI Agricultural Single Cell Workshop, just immediately prior to the LGBT Agricultural meeting. In this meeting in Orlando, Florida, it will bring together experts from the single cell field to discuss and develop the relationship between single cell technologies and agricultures. The second meeting I would like to highlight is the Gordon Research Conference on Single Cell Approaches in Plant Biology, which is happening in August 10 to 15 in Portland, Maine. This conference focuses on advancing the frontiers of science through the presentation of cutting-edge and unpublished research. I think these are really great opportunities, and if you can, I highly recommend to join these conferences in person. And just one last announcement before we dive into today's topics is if you have any questions, please write it into the Q&A box in Zoom, and we will try to answer in real time. But if you cannot get your question answered, you can also write a question into the Padlet, which you can access through this QR code. And this Padlet is open also after the webinar, so you can continue to discuss different questions and topics related to these and previous talks. So today we will have 2 presentations, and the first one will be from Professor Keiko Torii, and it is my pleasure to introduce her. She is from University of Texas, Austin, where she is a Johnson & Johnson Centennial Chair in Plant Cell Biology. Since 2011, she is also a principal investigator at the Howard Hughes Medical Institute. Professor Torii's work is truly outstanding, focusing on cell-cell communications and its role in plant development, especially focusing on stomata development. In her talk, Professor Torii will talk about cell states during development. Professor Torii, please.
Okay, can everybody hear me? Let me share the presentation.
Yes, we hear you.
Good. And let me put the PowerPoint. Okay, thank you so much, everybody, for this invitation. I don't myself study plant cell atlas or single cell biology myself. So this is a great opportunity for me to learn the community and learn about what are the exciting topics and what are the outstanding questions. So I really appreciate this opportunity. Thank you so much, everyone. So I start off with the question of what are the cell types and what are the cell states? And in the epidermal system of the plant leaf, which my group studies for many years, of course, we have this pavement cell, this highly cuticulated epidermal cell types that prevents plants from desiccation, pathogen entry, UV damage, and etc. But the plant epidermis also differentiate stomata or singular stoma, the paired guard cell that can open and close to adjust the gas exchange, but to protect the water loss when plants are under drought condition. But are they these different cell types, or are they a terminally differentiated cell state? Because developmentally, they came from the same initial cell called a protodermal cell. So initial cell, the protodermal cell, could enter into asymmetric stem cell-like behavior, which reiterate many times. It generates 2 daughter cells. Smaller one is called meristemoid, and the larger one we named stomatal lineage ground cell, or SLGCs. And the meristemoid can reiterate these asymmetric proliferative divisions for a couple of times, but eventually it loses the proliferative states and transition into differentiation and generate this stoma paired guard cells after the single symmetric division, at which point its sister cell, SLGC, could differentiate into the pavement cell. But it can be much more plastic that this SLGC can enter secondary asymmetric division to make a secondary or even tertiary stomata as well. So the cell types in the final stage, but during the process, they share the same trajectory or the same ancestry. So they are different state of the cell towards certain type of differentiation program as well. And these steps are regulated by the basic helix loop helix transcription factor heterodimers with species that initiate and promote proliferation, mute that promote differentiation, and the pharma that leads to the terminal differentiation, heterodimerizing with the paired BHLH protein named screen. And my work and the others has shown that peptide cell-cell communication mediated by the receptor kinase complex, including erector, too many mouths, and so forth, through MAP kinase cascade, is critical to enforce which cells during the two-dimensional differentiation of the epidermis will become stomata and or the pavement cell, so that the plants as a leaf can have a best fitness for gas exchange trade-off with the water loss. So because this system is kind of clear that these BHLH proteins regulate so much sharp cut of the cell state transition, we can see easily by genetic perturbation to convert the cell type from one to the other. So this is wild type developing leaf epidermis. You can see stomata or meristemoid that is dividing asymmetrically. And because of the cell-cell signaling and the polarity program, the stomatal distribution is even. But by treating one genetic mutation, this is SCREAM-D, you can convert everything into stomata, or vice versa, everything into pavement cell, this is speechless, and because it cannot breathe, this is a lethal mutation. Or you can even convert everything into stomatal meristemoid. So earlier time of non-single cell biology, that this sharp genetic conversion of the cell types or cell state allowed us to profile which type of genes are specifically expressed, for instance, in the stem cell state versus gut cell state or pavement state, so forth. So these are microarray era, but still be able to profile genes enriched or specifically functioning in the meristemoid, such as polar. But more recently, of course, technology advanced, and even though we are not really technologically savvy people, we start using those to really understand what is happening during the cell state transition. So in this case, we use the intact-based enrichment of the specific cell state during the stomatal differentiation and doing attack sequencing to see how the chromatin accessibility changes throughout the Arabidopsis genome. And we found, for instance, there are a group of regions that become open during highly proliferating stem cell-like state, but closes upon differentiation. And we identified the new regulator, BPC proteins, that works together with the differentiation factor, mute, to allow this transition at the epigenomic level. But more recently, of course, everybody knows that single cell technology is kicking in. And you all know this much better than I do, but this is from Jenny Rushinova's group's data that's showing stomatal cell types or epidermal cell types. You can do single cell RNA sequencing and map the trajectory from meristemoid proliferating state to differentiation state. But there are many outstanding questions. For example, what are the heterogeneities among them? Like, are they mature GCs or younger GCs or late meristemoid or early meristemoid? How heterogeneous in each cell among those populations? And the plasticity is another important question. How could what is the point of no return? Is this really the mute expression or something else? How flexible these fate and the transition can be? And of course, there's a question of progressive fate acquisition because meristemoi, as I mentioned, divide asymmetrically few times before commit to final differentiation. And not just doing a fancy analysis, some simple observations can also provide great insight. For instance, this is the early meristemoid just divided asymmetrically, and this is the later meristemoid that divided at least 1, 2, 3 times. And you can see there are some difference actually in size and so forth. And for example, recently we reported that along the asymmetric division cycle, the cell cycle time is linked to the cell size, which kind of makes sense. And recently, Dominic Bergman's group published this beautiful story that actually the size difference of the meristemoid is tightly linked to the fate difference. So smaller the meristemoid at the birth, more likely that they differentiate into the godmother cell and then god cell. So many things can be done as a combination of the profiling as well as very careful observation, potentially microscopy and very tedious counting and so forth. So today's talk, I would like to go step and try to address this question number 4. So how does cell state, is there any cell state specificity to the response to environmental, immunity, or metabolic signals, including plant hormones and small molecules? So this is kind of serendipitously getting into this field through my long-standing, decade-old by now, collaboration with the synthetic chemist at Institute of Transformative Biomolecules at Nagoya University, Japan. So together we initiated a very large chemical biology or chemical genetics screen to look for the small molecules that impact stomatal development in a different ways. And we got a couple of interesting ones. 2 has been published recently. One of them, KC9, which is a small molecule. This one increased the number of stomata and cause stomatal clusters. So that's the talk of today for the rest of my time. And this project was done by my talented former postdoc, Avid Herman, and my current senior scientist, Krishna Separ, the structural biologist, as well as a bunch of teams from UT Austin, as well as in Nagoya University. We also had some interesting compound published recently that reduced the number of stomata and more specifically triggers the stem cell arrest or meristemoid arrest. And this is also done by collaboration with both groups in Nagoya and here, including Ayami Nakagawa Sungi-han from Nagoya and my grad student Heimin Seo and senior scientist Krishna. So we'll talk about this KC9 story and I'll try to converge into how could this come into addressing the question of cell type specific response. So this question emerged from the bigger context of receptor kinase signaling, that what are the signal specificities between development and immune response. So it's been a many years a conundrum sort of questions because stomatal development pathway and many different pathway, including immunity pathway, is sharing the component other than the ligand, EPF, coming from plants, or flagellant peptide coming from bacteria, perceived by FLS2 immune receptor or erector family. And of course, the output, the downstream transcription factors are different. But if you look at the receptor partners and the immediate next component to MAP kinase cascade, they are completely shared. So this raises the question historically and still having a question that what makes the signal specificity between development and the immune pathway. So a couple of years ago, there was a paper published reporting that the antagonistic inhibition between the stomatal signaling pathway versus the immunity pathway maintains the signal specificity. And this occurs at the level of MAP kinase, this upstream MAP KKK, either YODA for stomatal development pathway, YODA is also MAP KKK4, or the MAP KKK3 and 5 for immunity pathway. However, more recently, there are 2 papers published. which saying, wait a minute, YODA and MAPKKK3 and 5 actually are redundantly functioning in both stomatal development and the immunity pathway. But if this is the case, it's coming back to the original question of then what determined the signal specificity. So I mentioned that through the large chemical library screen, we got a bunch of small molecules that increase the number of stomata. And this is one of them, KC9, which increase stomatal lineage cells as well as mute-expressing cells. Cells commit to differentiate into stomata. And this KC9's effect is dose-dependent. So more dose, I mean, it's saturated at some point, but more dose, it's increased the number of stomata. And we know that this phosphate residue as well as this naphthyl aryl residue is important because if you destroy here charge, then it's not bioactive anymore, does not increase the number of stomata. And if we make it too large aryl residue here, it also becomes biologically inactive. So we start wondering how could KC9 causes this excessive stomatal differentiation and the stomatal clustering, and we quickly be able to narrow down it's falling into inhibiting this erector-mediated signal transduction at the level of MAP kinase. So we did extensive application of the chemical onto all sorts of repertoire of stomatal development mutants. So we skip that. Sorry, Avid, he did the work. So I'll show that this KC9 is a MAP kinase inhibitor, especially MPK6 inhibitor. So a couple of years ago, we solved the crystal structure of MPK6, which we used to model the binding. And the KC9 seems to bind to occupy the ATP binding pocket of MPK6. And KC9, but not its inactive analog KC9-3, can inhibit the MAP kinase activity in a... Okay, and we also show that KC9 binds to MAP MPK6 as well as its dominant negative version. But if the kinase is constitutively active, its affinity or the KD value goes up, so affinity goes down, means KC9 is not a good inhibitor. If the MAP kinase is pre-activated, KC9 has a limited capacity to inhibit it. That's kind of important portion. And we further show it's not just the kinase activity, but the binding of KC9 to MPK6 prevent or inhibit the MPK6 binding to its downstream target screen. So you can see that in a BYFC here. The screen binding with MPK6 got decreased upon KC9 application. So it also inhibit the binding of MPK6 to downstream. Therefore, downstream screen and the speeches become crazy active to make more stomata. But then we got a question of MPK6 or MPK3, MPK6 regulates so many process of environmental stress and the immunity defense response. So if KC9 is the MPK6 inhibitor, it should inhibit many different pathways beyond stomatal development. So to address that, we collaborated with Seisuke Kimura and Riboshan, and we first did the RNA-seq analysis and did a self-organizing cluster analysis to see what kind of genes are upregulated or downregulated upon KC9 treatment. And the result is pretty clear. Of course, stomatal development sort of components, genes are upregulated by KC9. But if you look at the downregulated clusters or GO categories, they are overwhelmingly defense response, immune, abiotic response, all sorts of osmotic stress, salt stress, all sorts of non-stress signaling pathways mediated by MPK6 and MPK3. And we also show that KC9 can attenuate, even though very mildly, the FLAG22 triggered immune signal activation as well. So this led us to start looking more carefully how immune signaling pathway regulate stomatal development. So getting into more cell type or cell state specificity. And the result is quite shocking. So I'll try to walk through as much as I can. So here is the, again, the EPF erector-mediated stomatal development pathway. And here is flagelline peptide FLS2-mediated immune response pathway. So this is a wild-type leaf epidermis. And if you apply KC9, you get many stomata that are clusters, as I showed you before. If you apply flagelline 22 peptide to wild type, it really doesn't do much in a stomatal development. Their shape kind of become funny, but they look pretty normal. But surprisingly, if you apply KC9 and the flagelline 22 peptide together, then what we see is there's no more increase of stomatal numbers, no more clustering. So somehow FLAG22 peptide prevent KC9's action to inhibit inhibit species to make more stomata. And this is due to the proper signaling perception of the FLAG22 peptide by FLS2 receptor, because if you do the same experiment with FLS2 mutant that lacks the immune receptor, then the flagelline peptide, FLAG22 peptide application has no effects, cannot prevent KC9's ability to increase stomata. So this is really what's happening is through FLAG22 perceived by the FLS2 signal transduction. So here's the answer. So in the presence of KC9, the immune signaling pathway somehow feeds into stomatal development pathway. And what's happening is, so we start looking into the progressive loss of function of erector receptor kinases. There are 3 of them. And if we knock out 2 out of 3, what we found is flagelline 22 peptide alone can decrease the number of stomata. So you don't really need KC9. KC9 can have them back up actually in this case. So this indicating that it's not the weird phenomena because of the small molecule KC9, but if some of the part of the receptor kinases for stomatal development are missing, then immune pathway can interrogate to repress the number of stomata or activate the signal to repress the number of stomata. So KC9 is simply mimicking the partial loss of stomatal receptors. But then we got really interested in what happens to what cell state this is happening during the epidermal development, because all the studies we did so far is we looked at the day 7 or something like that. So Avid designed the experiment that throughout the stomatal development, he added KC9, this compound that increased stomata, and then the day 1, day 2, day 3, day 4, each day, he added on FLAG22 peptide and see when during the development or the germination, this is a cotyledon development, that stomatal development can be shut down by FLAG22 FLS2 immune signaling pathway. So if you add FLAG22 peptide from day 1, of course, you don't have stomatal increase. And if you just add flag peptide at the last day, of course, it's just a KC9 effect. You see many stomata in the clusters. What about in between 2 to 6 days, right? And the result is quite striking because we saw something new at day 4. At day 4, when we add FLAG22 peptide at day 4 and let them grow together, then we saw this kind of arrested small meristemoid-like cells, like gather together, like what's happening? And the first question is, are they meristemoid or something else? And the result is they are something else, because unlike normal stomatal development trajectory, these small cells seemingly looks like a meristemoid and others. They no longer express stomatal lineage markers or stomatal differentiation marker. So they kind of got shut down. They're small in dividing cells and they shut down. So that means they are withdrawn from the stomatal lineage identity. So we believe that this actively dividing stem cell state or meristemoid state is most vulnerable to the signal interrogation or signal invasion from immunity pathway that activate the same MAP kinase cascade. So combining everything together, so here is our current model of how could signal specificity is maintained and how can it be broken. So if the receptor, the activable components that can be activable by the proper signaling ligand receptor interaction is fully available. In short, if the homeostasis of MAP kinase cascade is such that they are ready to be fully activable by the receptors, then during such cases, two pathways, development and immunity, can maintain signal specificity. However, if you weaken the MAP kinase activity by KC9, the small molecule, or if you weaken these activable receptor pathways by removing 2 out of 3 receptor kinases upstream, erector family, then the FLAG22 FLS2 mediated activation of the MAP kinase can cross-regulate the stomatal development. And in short, having the flux and the homeostatic state of each pathway is critical to maintaining the signal specificity. But then coming to the cell state specific signal cross-regulation, what are the outstanding questions and what can we use single cell or the cell atlas approach to understand this phenomena much better? So the questions such as, is this cross-activation specific to FLAG22 FLS2, or are they also shared by other signaling pathways that converge into MPK3 and 6? There are so many, as I mentioned, abiotic stress and different kind of immunity and so forth, and different kind of development as well. And what is the key? Is the heterogeneity of the small proliferating cell important? Or is that the signal strength important? Or is the expression pattern of the receptors important? And if you look at the single cell available data, of course, these receptors for erector and its paralog, or FLS2 and similar immune receptor EFR, they're expressed in a different cell types or maybe cell state. And that could combinatorially impact the cell fate decision during the cell state transition and the cell fate decision during epidermal development. And what looking forward, I'm not getting to explain all the data or in this published paper from Jen Shendua and Tapnell's group. In animal single cell people, I start doing single cell receptor kinase or kinome chemogenomics type of approach. They're combining everything, and I don't plan to combine everything, but some of the essence is very interesting to learn and adopt. So in this single cell general perturbation, essentially they are using CRISPR library sort of perturbation of the entire kinome and put all sorts of the kinase inhibitors and the other type of inhibitors to try to dissect out single cell or the cell state specific response to signal interrogation and the signal perturbations. So something like that can be easily adopted to plant cell atlas group, and also coupled with careful live imaging and so forth. Maybe need a good biosensors for MAP kinase activation to really understand how cell can use the same signaling component, yet able to achieve specificity to generate perfect epidermal tissues. And having said that, I would like to thank all the members of the Torii lab and the collaborators and people from Nagoya University and synthetic chemists who made all of this exciting journey possible. And we are currently looking for lab managers, and we are always looking for postdocs. And again, I'm not a single cell expert, but if any of you have expertise and interested in joining the team, I sincerely appreciate. Call me. All right, thank you so much for your time, and I'm happy to answer any questions.
Thank you very much, Keiko. It was a great presentation. Just again for the audience, if you have any questions, please type into the Q&A box in the Zoom so we can ask. There is one question from Mario. Is KC9 providing an adaptability response to plants challenged with a pathogen, for example, botrytis? In other words, does the crosstalk has an ecological response?
Yeah, that's a great question, and we would like to definitely getting into that. So, one obvious thing is that it's very well known from study from Shen Yang He and Libo Shan and many others, Cyril Gipfel, stomata and Jilke Roberstack, that stomatal immunity, that stomata shut upon detecting the FLAG22 peptide to prevent the entry into the internal mesophyll tissue, right? So the question is, that's a short-term response because stomata opening and closing is fast. But in a longer term, that's reducing the entry points or the reducing the number of stomata really make a difference in the longer term to preventing or limiting the pathogen entry? So that's a very exciting question, and we would like to test. And we actually tried. We tried a Arabidopsis seedling treated with a Pseudomonas syringae DC 3000, but we kind of killed the plant. So I think we are not. It's more like the rate limiting step here is our inability or the inexperience to design proper experiments. So I would definitely like to have a collaborator or try to do some experiment as well. Thanks.
Thank you. There is another question, and after that, I think we need to go to the next presentation. So this question is from Marketa. Do stomata develop the same way in cotyledons and in true leaves?
That's a great question. It's not necessarily identical. So because the cotyledon initiate during embryogenesis, so they are pre-patterned. So you can see the patch of mute expression during the species expression during embryogenesis. But once they become mature and desiccate and become dormant and regerminate, they can reinitiate the pattern. So in that aspect, they are similar. So if you treat with EPF2 peptide, for instance, of the germinating cotyledon, you can completely block stomatal differentiation. So there are difference of completely de novo versus pre-patterned during embryogenesis, but the pre-patterned stomata cells are still plastic. They can respond to external signal to inhibit or increase the number of stomata. So they are not fully committed. So they can be a cotyledon versus leaf stomatal development could be a good system to understand how commitment or plasticity are specified. I think some people are studying that in Germany. Yeah. Thank you.
Thank you very much. There is a few more questions in the Q&A. If you would like, you can answer in the Q&A by typing an answer, or we can bring it to the Padlet. But I think it's better to move on because the time is a bit limited. So thank you very much, Keiko, for the presentation.
Thank you.
The next speaker is Dr. Justin Walley. Justin finished his PhD in 2009 at UC Davis, and since 2015, he's an assistant, then from 2020, an associate professor at Iowa State University, Department of Plant Pathology and Microbiology. His lab focuses on proteomic and biochemical approaches to understand the underlying mechanisms of plant development and environmental responses. His talk is titled Proteomic Approaches to Define Cell Types and States. Justin, the stage is yours.
Thank you for the kind introduction and the opportunity to speak here today. My screen up now?
Yes, all good.
Okay. So I think there's been a number of fascinating talks in this series, and I think I'm the odd one out that's focused more on technology than biology, so bear with me. But today I'm going to discuss kind of different approaches to use proteomics to understand the proteomes of single cells. And there's lots of different ways to go about this that have been developed primarily in mammalian systems. And there's been a few examples now in plants that I'll highlight. But again, I'll kind of show the breadth of the technologies that are able to be used and kind of some strengths and weaknesses for each of them. So there are trade-offs with each different technique. But coming back to this idea of cell state reminded me of discussions I had over the years with Steve Briggs when I was a postdoc with them, where we would discuss the idea of a proteotype, which was the proteomic state that kind of was impinged on by development, plant's genotype, and the environment to drive phenotype. So potentially this proteomic state also is what underlies cell state differences, and we can, discuss that at the end potentially. But why, the single cell RNA sequencing has taken off? Why is it challenging for proteomics to do similar analyses? And I'll highlight a few of those challenges. One is simply, two of them are biological in nature. So if you take these examples from mammalian cells, a given mammalian cell contains billions of protein molecules, but less than 500,000 RNA molecules. So you need 30,000-fold more counts to characterize protein molecules relative to transcripts. So you have to analyze a lot more stuff just to look at a single cell when you're looking at proteins. And there's also a much bigger dynamic range of proteins. So the proteins can be present at tens of copies per cell, while others are at millions of copies per cell. So you have this large 10 to the 6 dynamic range that is much larger than the RNA's dynamic range. And, quite intrinsically, you can't amplify proteins like you can RNA. So whatever amount you have in a cell is the amount you're going to have as the analyte. And to highlight this dynamic range issue, here's some data that Bruker generated for us a year or so ago. But what is shown here is just bulk proteomics of Arabidopsis seedling tissue. And in this analysis, 12,000 proteins were quantified. But if you rank order the proteins based on their abundance or signal intensity, 75% of the intensity or ions that we're analyzing come from only 32 of the proteins. So again, 32 out of the 12,000 proteins quantified account for 75% of the signal. So much of what you're detecting are these really abundance proteins. And it's being able to get into these lower abundance range. It's, I think, where we're interested biologically. So to kind of orient you through how proteomics is done in general, I like to break it into 3 parts. So you have sample preparation and separation. So with proteomics, most of it is bottoms up. So you take proteins, digest them into peptides. This would be analogous to shearing RNA for RNA sequencing. And then these peptides can be manipulated in different ways, but ultimately they're separated by HPLC and ionized into the mass spectrometer. There's lots of different forms, and I'll highlight the mass spectrometer forms. And as I go through proteomics, I'm going to focus on 2 aspects going forward. One is the sample preparation part, and the other is mass spectrometry choices you can make. But I want to just highlight with the sample separation. So for all of the single cell proteomics I'm going to be discussing, folks use one-dimensional low pH reversed phase fractionation. So this interfaces the HPLC separating the peptides with the mass spectrometer. If you're doing bulk proteomics, you can add other dimensions of fractionation to increase proteome depth. There simply isn't enough sample in a single cell to add these other dimensions of fractionation. So again, everything that's used is one-dimensional fractionation, and folks play with the flow rates, but there's not a lot that's changing between different approaches for single cell proteomics with that. So I'm not going to touch any more on separation. And I mentioned, mass spectrometer is a key part of this proteomics. There are hundreds, if not thousands, of different mass spectrometers out there. For proteomics, there's 3 main mass analyzers that are used. All the single cell proteomics work you'll see incorporates mass spectrometers that have a quadrupole. And this is a filter essentially to let specific ions with a mass to charge ratio that's set to go through to the detector, and then it can scan through. So again, most of them have these quadrupoles. And then the 2 other mass analyzers that are used, and more recently, are these time of flight. So it measures how long it takes an ion to get from point A to point B. So these have very fast scan rates and medium resolution in terms of their mass resolution. And then the Orbitrap is the other predominant mass analyzer. So it has a medium scan rate, so you don't analyze as many ions, but it has higher resolution. So these 2 are often used interchangeably for different reasons. And then within mass spectrometers, there's 2 main ways to acquire ions or information. So the first and the kind of classical way is data-dependent acquisition. And with this, depending on what data is present, and by data I mean what ions are present at any given time, the mass spec decides which ions to sample. So you can see here in this MS1, this would be an ionized intact peptide. The mass spec sees what ions or peptides are most intense, and then selects those ions mass to charge one at a time for fragmentation to get this MS2 or product ion spectrum. So this product ion spectrum is what identifies the peptide. So again, depending on what's present at a given time, those ions get sampled for MS2 and identification and quantification. And this leads to stochastic sampling of these precursor ions. So not every ion is going to be the most abundant in the different mass spectrometry runs. So you get more missing values, meaning you identify something in one run, but not the next with data-dependent acquisition. And in kind of a method that's been taking off in the last several years, especially for single-cell proteomics, is data-independent acquisition. And here, the MS1 provides a snapshot of what precursor ions are present, but no information is used from that other than what is there. So it's not selecting any of these ions per se for fragmentation. Rather, there are these predefined isolation windows that are cycled through every run. So it's just cycling through these same isolation windows. No data is used to decide which windows are used in any acquisition. And because of that predefined window selection, you're isolating the same masses every time. And that leads to less missing values between runs. And this is taken off because of the faster scanning mass spectrometers and computational improvements. Another feature I want to talk about is ways to multiplex. A common way, especially with single-cell proteomics, are these isobaric tags or tandem mass tags in this case. And they're used with data-dependent acquisition for quantification. And what they have is this mass group, and you can have an amine reactive group on it. So this attaches these labels or tags to your peptides. There's a mass normalizer and a mass reporter group. And so in this case, when it's intact, all of your labels or tags have the exact same mass. That enables you to label each one of your sample, or in single-cell proteomics, each cell with a different mass tag. You can combine them into a single tube and then analyze them by mass spectrometry. So that MS2 step where you add energy and fragment your peptides into product ions, it also cleaves off these mass tags. And now you have your normal part of your spectrum for identifying the peptide that is present, but then you have these mass reporters that are free and tell you the amount of each peptide that was present in any given sample. And so this kind of relates back to single-cell proteomics because there's a number of challenges that need to be overcome. I think the biggest challenge is throughput. So these label-free approaches have lower throughput, and throughput is really limited by mass spectrometry analysis time. So you can see for number of single cells you can analyze per sample is low. So with label-free, again, it's one cell at a time. Less cells can be analyzed in a given day. And then these multiplexed tandem mass tags with data-dependent acquisition have lots of multiplexing tags available, so you can get more and more single cells analyzed per sample or per mass spec time. And one approach that's kind of taking off and working to be developed are ways to multiplex with data-dependent acquisition. These right now are limited by the number of tags you have. But to give you an idea, the throughput is now possible. Single cell label-free approaches are on the order of 30 to 40 cells per day, so not very high throughput. The multiplexing with DIA is in the 100 or so cells per day. And then now with 32 plex tags, tandem mass tags, Nikolai Slavlov's group has reported analyzing about 1,000 single cells per day. So throughput is getting much better. There's a little bit of a trade-off with depth in terms of number of proteins detected, but I think that throughput is really key for doing larger scale studies. And then there's another challenge with proteomics is sample handling. So this is irrespective of the mass spectrometer now, but we're dealing with low protein amounts in a single cell. Just isolating cells is a challenge. You need methods that can remove or eliminate the use of detergents. It's harder to remove the detergents from the sample when it's present with a single cell. So the ways to manipulate cells without detergents are key. And then because of the low sample amounts, we really need to work on reducing volumes of the samples, because as samples get diluted, peptides in low abundance really will start absorbing really strongly to the walls of the tubes that we use and you lose sample that way. So again, it requires miniaturization of the volumes used to handle. And there's a few main approaches that are used. I'll talk more using about this MPOP method, where you're doing sample manipulation in 384 well or some number of well plates, typical, PCR plates. These are in the microliter range of reactions. So we work to miniaturize, but you're still in the microliter range, but it doesn't require any of the more fancy robots, and I'll discuss cell line 1 in a second. And then what's kind of been developed in a number of different approaches, they have different names and different ways of doing them, but are fabricated glass slides that have etched nanowells in them. And then this cell line 1 robot is used to dispense really small volumes into these arrayed nanoliter wells. And you can see now we again are in the nanoliter range. You can multiplex with these as well. And the cell line 1 instrument can now process, thousands of cells per day. This doesn't include mass spectrometry time, but you can get samples ready in a really high throughput manner. So these again, miniaturization with robotics, the cell line 1, here's what these slides look like, where each one of these little dots is a different nanowell. And this machine or instrument can move liquids into these little droplets that are formed, and you can do your protein, extraction and digestion all in the same well. You can array these again into your 384 well plates to put in the HPLC to separate. Or there's some groups have developed their own custom robotics, like this nanopots that you might read in a number of papers, where the slide is interfaced directly with the nanopots autosampler. And what these slides do in addition to reducing volumes, is this nice approach by the group that developed the nanopot system was a way to do RNA sequencing and proteomics on the same exact cell. So this is called nanosplits. And the idea here is you form the droplets with a single cell in them, and you essentially sandwich two of these plates together. And half of the cell goes on one plate and half of the cell goes on the other. And they benchmarked this that it really reproducibly does split at a very precise volume. And there you can take and do RNA sequencing on half of the cell and proteomics on the other half. And with their approach, they're able to quantify about 6,000 transcripts and 3,000 proteins per cell. And they asked a number of questions, including how well does the transcript abundance and the protein abundance in single cells compare? And similar to bulk, you get a moderate Pearson correlation between RNA and proteins. But I think this opens up a number of ways to ask kind of more interesting biological questions about what's going on in cells. And then, those were kind of some methods that are used, and I wanted to focus on, the plant biology now. And the same group that developed nanopots also has a very nice preprint out where they use label-free proteomics on Arabidopsis mesophyll cells collected from leaves that had experienced drought or not. And they were able to use, again, this cell line 1 and nanopots to do some really nice proteomics. And I won't steal their thunder, but I encourage you to read through this preprint. There's some nice biology in there. And then for the last part, I'm going to talk about work my group did together with Trevor and Philip to look at single cell proteomics of two predefined populations within the Arabidopsis root, single cells from the cortex and endodermis. And I mentioned, we use this MPOP method, which is in the microliter volume range. We also use tandem mass tags. You'll see this SCOPE acronym frequently in single cell proteomics papers, but it's a method for multiplexing with the tandem mass tags. And the idea here is you have your single cells that are arrayed, in this case, in a 384 well plate. You can process them and add a specific tandem mass tag to each single cell. Then you have one of the well in each multiplex set is labeled with 5 cell equivalents of peptides. So this helps reference and normalize across multiple multiplexes of these samples. So when you're doing larger studies, you need to normalize from mass spec run to mass spec run. And then we used, and some groups do, some groups don't, this idea of a carrier. So here, some number of cell equivalents of peptides, say 100 cell equivalent of peptides, is labeled with one of the tandem mass tag channels. And what this does is when you mix all these together and analyze them by mass spec, again, the mass MS1 isolates all the peptides with data-dependent acquisition. When you fragment it, you release these reporters, and now you're able to quantify the intensity or the protein's intensity from each of the single cells. You have the normalized tandem mass tag channel abundance in one of the wells, and then the carrier helps amplify the signal. So it's not used for quantification, but it helps used for detection, and then we process the single cell data. So how this all worked together is Trevor and Jay from when Trevor was in Phillips Labs, sorted these single cells from lines with endodermis and cortex markers into single wells on a 384 well plate using FACS. Christian and my group worked to use this Opentron's OT2 robot to do all of the manipulations in terms of cell lysis, protein digestion with trypsin, tandem mass tag labeling, and then pooling all of these. So this was a fair amount of work to get this up and running. But what we're able to do is have single cells, that reference and carrier, and analyze them by mass spectrometry. And in all, we processed about 770 single cells. You can see for this point here at the protein false discovery rate, we're identifying about 3,700 proteins total across these cells at about 1,100 proteins per cell. We did a number of filtering steps to look at cells and proteins that had a low coefficient of variation and high precision in their quantification. And the data I'll talk about today focuses on 3,200 of these proteins that were analyzed across 81 of the cells, so 33 endodermal cells and 48 cortex cells. This filtering step eliminated a number of the cells. And I want to highlight here, this work was done on a really old mass spectrometer. So the technology, the Q-Exactive Plus instrument was released, I think, 14 years ago. So this wasn't the newest and greatest mass spectrometer, and we're still able to get pretty good identification in terms of proteome depth. Obviously, with newer mass specs, these numbers can increase. But even with this, we're having really nice biological separation, as you'll see, of the cell types. So some things we looked at of what kind of proteins we're detecting. We compared this from Rachel's paper in Developmental Cell a few years ago, looking at the single cell RNA-seq from these, across the root, and we focused on cells from these two cell types. And we simply did is rank transcript abundance of the single cell RNA sequencing data, and then color-coded if we detected that gene's product as a protein as well. So did we detect a transcript and then also as a protein? And you can see the proteins we detect range from very low to very high abundance of transcripts. So we're getting the full range of transcripts abundance detecting the cognate protein from that gene. But of course, there's a bias towards detecting transcripts that are higher in abundance. And another thing we looked at is, can we detect proteins folks commonly associate with signaling, transcription factors, and kinases? So here it's protein abundance ordered across our single cell proteomics data. And you can see we detect both transcription factors and kinases ranging from high abundance to low abundance. And in total, out of these 3,200 proteins, we're quantifying about 175 kinases and transcription factors. So we're getting a fair amount of these types of proteins being detected and quantified. And that leads to the question, did we find anything biologically meaningful? So one of the analyses we did is just look at differential enrichment of proteins between the cortical and endodermal cells. And here showing these MA plots where you have intensity plotted against fold change. So stuff that's up here is enriched in the cortex and stuff that's down here is enriched in the endodermal cells. And what we can see are proteins like auxin response factor 5 being enriched in cortical cells. It's, a protein that's known to drive cortical cell division. And, we find a few other markers as you'd expect. Similarly, for the endodermal cells, we find proteins like CASP1, which drives Casparian strip formation in the dermis, being enriched in the endodermal cells. Similar NPY2 and RBOHD are also where you would expect them. So we're finding markers where we would want them, and now we have a number of other proteins that might be interesting to look in for driving epidermal or cortical cell fate. And then finally, we wanted to ask, could we actually differentiate the proteomes of these two cell types using different analysis techniques? So here's two-dimensional reduction techniques, PCA and UMAP. We performed it on those 81 cells that had robust quantification, and you can see by color coding, the cortical cells and the endodermal cells are separated by both of these methods. And then similarly, if we hierarchically cluster each of these cells, proteomes, we see separation by and large from the endodermal and cortical cells. So I think this really highlights that we're detecting not just simply high abundance housekeeping proteins that are the same in all cell types, but we're detecting proteins that can differentiate, the proteomes of different cell types. So looking forward, I mentioned that we threw away a number of the cells, actually most of the cells, and we believe that this is largely biological, driven by cell size differences along the root gradient and also cell cycle differences. This cell size impact has been noticed in mammalian studies as well, where they're able to use histone proteins to normalize as a crude marker for cell protein content. Challenge in plants and tissues like the roots they're undergoing endo-root duplication, and I believe this is going to make histone proteins poor normalizers for cell size. So one of the areas we're working on currently is, whether it's a new FACS sorter we have on campus or the Selenium 1, you can bin and actually image each cell that you're acquiring. So we want to do single cell proteomics on cells of different size and find proteins that might be ways to normalize for total content. Other ways of sample methods are progressing rapidly. I've mentioned this nanopots and Selenium 1 are both custom and commercial sample processing systems. These are active areas of development. There's also these TCAN Unos, which are good for nanoliter dispensing that we're working to help reduce volumes as well. But I think the big thing that's got to shift is accessibility. So this requires lots of instrument time, especially if the cell or samples are not multiplexed. So the mass spectrometer accessibility is an issue that needs to be worked through. And the need for increased coverage or depth of the proteome is realized. But I think again, the throughput is the largest bottleneck currently. And newer instruments from both Bruker and Thermo are rapidly increasing both of these. So now, depending on the cell type, there are reports of up to 5,500 proteins per cell, analyzing about 80 samples per day. That's without multiplexing. So lots of rooms for improvement, and these newer instruments will help. And then finally, I think opportunities for single cell proteomics. I see a couple that are, kind of readily available, or one that's readily available, and that's using it to explore heterogeneity within single cells. But in particular, how can we address how individual cells of a given cell type respond very differently to stimulus? So whether this is hormone or abiotic, like salt or something like that. But again, how do individual cells respond differently? And can we pair this with RNA sequencing in a higher throughput manner to really get at predictive regulatory networks and mathematical modelings of what's going on in these cells? And with that, I'd like to thank especially Christian and my group who did most of the work on our end, as well as Trevor, Jay, and recognize Philip for all his great contributions to the field and helping make this work possible. So with that, I'll finish up.
Thank you very much, Justin. It was a great talk and great to see some orthogonal method to the most common single-cell RNA-seq techniques. There is one question from Juan Jose. To what extent protein turnover/dynamics can impact your profiling and need to be taken into consideration when designing your workflows?
Yeah, so I think, it's the same issue with RNA sequencing. Both RNA and protein are dynamically turned over, and both of these methods are going to be looking at steady state levels. So with this, I think same as RNA, it's working as fast as possible. So that's one of the nice thing about some of the robotic systems is where you're collecting cells within 5 or 10 minutes and then lysing them in conditions that largely will inhibit the proteasome and other quality control processes that are degrading proteins. But that's always an issue with both RNA and protein.
Great. Thank you. Just another question from Guiming. Thanks for the presentation. Would there be enough materials in single cells to enrich for the phosphoproteome? Can we add more carrier cells for this purpose?
Yeah, so the carry, so one way to do it, so there's two concepts of carrier, and one is the way I described here with the tandem mass tags, and you're adding actual peptides. For some of the data-dependent acquisition methods, folks can analyze in a separate mass spec run, like 40 or 100 cells, and then use that in the protein search engines as one of the, not a replicate, but as one of the samples in that search. And that helps do identifications across runs. So you can boost abundance of lower abundance analytes that way in terms of their detection. So presumably you could enrich for phosphopeptides and also use that as a carrier, either in the tandem mass tag strategy or in the data independent acquisition. And then I believe in one of the newer papers describing Thermo's kind of high-end mass spec, the Astral, they didn't do any enrichment, and they're detecting, I think if the number is right, several hundred phosphopeptides. So without enrichment, they're just sampling enough ions to see some of the high abundance. But I think any terms of like actual analytical chemistry enrichment will be difficult at a single cell level.
Thank you. And just a more general question to answer the webinar's main questions from me is that what do you think using the single cell proteomics techniques, how you can redefine, modify, or change definitions of cell types or states?
I've heard, and discussed this with Ken and heard him talk frequently, and I often think about this and I still don't have a good answer, but I think it's a very interesting question. I mean, I think knowing what the cell states or the proteomes are is the first easy look at something and on the protein side, but actually understanding what's driving what is a big, more fundamental question. So I don't have a great answer for that.
Thank you. That's fine. And I can ask the same from Keiko from your presentation, just to wrap it up and to try to answer the webinar's questions. That you showed these, you mentioned cell states rather than cell types, so different states of the same type development and the cell-to-cell communication. And what do you think how this cell-to-cell communication and different signaling pathways could be used or utilized to redefine or refine cell type and state definitions?
Yeah, that's a fantastic question, and that's what we are kind of try to answer through my scientific career. So it's not a quick thing, but as you mentioned, it might be my personal definition, but when you think about cell type, it's like defined cell type, either gut, stomatal gut cell, or pavement cell, or endodermis of the root, or cortex. But they are not a black and white. We see those snapshot of final stage that they acquired, because multicellular organism is division of labor, so they make a different cell type. But on the trajectory to go into that cell type, and you don't want to make too many stomata or too few stomata. Everybody knows from our mutants that how detrimental they are, right? So cell-cell signaling always have to kind of give them instruction. Wait a minute, don't go that way. Turn left. In a way, like a mom for telling my daughter, don't drive too fast or don't drive too slow, don't go to that street. They are always giving that, in a way, instruction, but that also changes given the situation of the plants should grow or plants should defend or adjust to drought. So in a way, everything is mixed together and integrated together. So it would be an interesting challenge to utilize this my type of reductionist approach together with combining more integrative, maybe using AI and as well as our patient eyes to look at the microscope and so forth, to combine to really understand what collectively allows cell or cell state to maintain plasticity so that as a mature organs, whatever final stage they make is always best fit or the best outcome of the given environmental circumstance. I think there's no simple answer, but I think many plant developmental biologists like myself is really excited that now we have many tools, whether it's a single cell proteomics like Justin is developing, or all sorts of a fascinating approach that I'm not a technology person. So something like Justin's talk, I'm like, wow, amazing. So I think it's really the time that those can be integrated with collaboration and scientists all together and really understand for making the best plant outcome in a coming environmental situation and environmental changes. Yeah, so thank you so much for this opportunity for me to share some of our work and learn about exciting latest technology as well.
Yeah, thank you very much. And due to that time, I think we should also wrap it up. So thank you very much, Keiko and Justin, for your great presentations and insights from these techniques and biology. And we will see each other on April 8th for the next part of the Plant Cell Identity webinar series, where we will hear from Dr. Jess Dickinson and Dr. Nemanja Vukasinovic. So thank you very much, and have a nice day.
Thank you.
Thank you.